Hello guys and welcome back to Excelsior Academy. So this is your last topic in the unit 2 of cell biology, that is the microbial physiology. And in this we are going to discuss like the growth of a bacteria, like how the yield and the characteristics, what are the strategies of the cell division and how the bacteria is going to respond to the stress. Okay, so this is like very basic actually. Here we are going to discuss the growth curve, right? And later on we'll discuss the mathematical representation of the growth curve, like how the bacteria is dividing. So we know that it is dividing when it is in the log phase, it undergoes exponential rise, right? So how that can be represented in mathematical way, that we will be studying. And after that, how the environmental factors is affecting the growth of a particular cell, like what happens when a bacterial cell is under starvation. Okay, what happens when the pH is extreme, or the temperature, or the salt concentration. So it's a very basic topic, and we'll be just going through all this thing. And still guys, if you have doubt, you can just stop me. Okay, this is like very basic. Okay, so the growth may be defined as increase in the cellular constituents. It leads to rise in cell number when microorganisms reproduce by process like budding or binary fission. So needless to say, everyone knows how bacteria is dividing. In the later, individual cells enlarge and divide to yield two progeny of approximately equal size. Now suppose if it is being asked, like to you guys, the cell is there and it has increased in the size. Does it mean that the cell is still growing? Yes, Akshaya. If I say that this is from here to here, see, when we say the cell is there and it divides in two, we say cell division is happening, cell reproduction is happening, right? So the particular bacteria is growing. Now, if I say that this cell has increased in its size, can we say still that the cell is growing? Yes, anyone? Chandani, what do you think?
Ma'am, I think no.
Sorry?
No, ma'am. It's not growing.
Krishita?
Ma'am, I missed the question part of it.
Okay, okay, you joined late, na? So I was saying that, so we know what is growth. So it is increase in the cellular constituents, right? So I was asking if a cell is there and it keeps on, increasing its protein concentration or whatever, and it is increasing in its size. Can I say that the cell is growing rather than dividing? It is just getting increase in size.
Yeah, it is growing, but it is not dividing.
Yes, yes, correct. So even though the cell is not dividing and it is increasing in its size or length, we say that the cell is growing. Okay, so this question was asked in one of the interview. That's why I asked this question. Okay, so growth also occurs when the cells simply become longer or larger. Okay, it's not necessary that every time the cell has to divide. Okay, if the organism is xenocytic, yani ki it is multinucleate, okay, in which nuclear divisions are not accompanied by the cell divisions. So the cell is growing, but the cell division is not happening, even though the nuclear division took place. So that's why, because of this, they have so many number of nucleus, okay, but only one particular cell is there. Now, it is usually not convenient to investigate the growth and reproduction of an individual microorganism because of their small size. So do not think that if the size of the cell is small, it is not growing. Okay, so that is wrong assumptions. So when we study growth, microbiologists normally follow the changes in the total population number. So there are certain bacteria which are like, small in size. So you cannot say that that particular bacteria is not growing well. Okay. Now, population growth studied by analyzing the growth curve of a microbial culture. So when organisms are cultivated in liquid medium, they usually regrow in a batch culture or a closed system. That is, they're incubated in a closed culture with a single batch of medium. So what does it mean? We say that, अगर जैसे for example, let's say that we are doing in a lab the growth curve. कैसे करोगे इसको? So usually what we say is that आपके पास यहाँ पे culture है, ठीक है? You have inoculated. So obviously the first OD what you get at 600 is going to be 0, right? जो blank रहेगा, यहाँ पे आपका blank क्या रहेगा? Media. Okay. So normally in the lab we use what kind of media? LB, Luria Bertani media, right? या कुछ लोग NB भी बोलते हैं. So either you can use LB or you can use NB, nutrient broth. Okay. Now, let's say that we have started this in the lab. So आप सबसे पहले क्या करोगे? You are going to take, okay, a small conical flask and 100 ml of media. You're going to prepare it, sterilize it, right? So sterilization means यहाँ पे आप autoclave करोगे. So I hope everyone knows autoclave, like 121 degrees Celsius for 15 minutes under 15 lb pressure. Okay, so this is the ideal condition at which the sterilization is taking place. Now, what is the next step? In the hood, okay, laminar hood, you are going to take the culture and inoculate into the media, right? Media में inoculate करोगे, then you are going to keep it on a incubator. Now, this incubator is also having the shaker. I hope everyone knows why shaker is required. Akshaya, why shaker is required?
For equal distribution of oxygen.
Yes, correct. The correct word is aeration. Very good. Okay, so then you're going to keep it over there. Now, when we do the experiment, it is said that, there are few people who take the readings at every 4 hours or, at every 6 hours. So that they get a complete idea. ठीक है? So एक तरीका ये रहता है कि you take it in a one single conical flask, and every time at every 4th and 6th hour you take the conical flask, go into the laminar hood, take the OD and keep it back. ठीक है? In order to avoid contamination. एक ये तरीका होता है. दूसरा तरीका क्या रहेगा? Like if suppose I decided to take the OD like for 8 readings I want. ठीक है? अब 8 readings है और 24 hours है, so it means what? At the intervals of 3 hours, correct? ये तो समझ में आया ना? 8 readings है and 24 hours में मुझे लेना है. So obviously it means at every 3 hours I'm gonna take the readings. I hope this is clear to everyone. इतना
Yes, ma'am.
math है ना? Okay. So 0th hour क्या होगा? The moment you have added the inoculation. ठीक है? So that should show 0 OD because it is almost your media only. अब आप क्या करोगे? 3 hours है, 6, 9, like that you keep on doing. ठीक है? तो एक तरीका होता है कि आप एक conical flask में ही reading लेते रहो. So what happens is like every 3 hours you have to bring the conical flask, you have to take it out from the incubator, put it into the hood and then take the reading, then again put it back. Here you have to maintain the contamination free zone. ठीक है? दूसरा तरीका क्या रहता है? You take small conical flask, ऐसे 10 ml का, ठीक है? And 8, you will take around 8. Since 8 readings I need, so I'll take around 8. So अब मैं क्या करूँगी? 10 ml में at the 0th hour, I'm gonna add the inoculation to all the 8. ठीक है? Now I'll label as 3, 6, 9, 12, 15, 18. ऐसे करके मैंने इसको लेबल कर दिया है. Okay. Now what I'll do is at the 3 hour मैं एक उठाऊंगी, सीधा रीडिंग लूंगी and I'll discard. without disturbing the rest. So I'm not taking the others from, outside. I'm not taking from the incubator, taking outside. Now, when the 6th reading is coming, I'll take the another one. I'll take the reading and I'll discard. Discard. Similarly, till 24 hours. So which is the best method do you think? The first method or the second one, where I have only 100 ml and here I have got 10 ml ka at least 8 chote conical flask. What do you think, which is the best method and why?
Second one.
Okay, I agree. Second one is the best method. Now why?
Because there will be no contamination of rest of the conical flask.
Yes, correct. So in the second, even though it looks very tedious job, like you have to, make it 10 ml and you have to sterilize it everything, then 8 times inoculation you have to do instead of one over here. But if you have to take it 8 times readings, right? So the chances of contamination is increasing in this case, because 8 times you will take this out from the incubator, open it, take the reading, and again keep it back. Here is like once you take out, take the reading and throw it. Second, take the reading, throw it, right? So this is giving you the best way where the chances of contamination will be very, very less. Okay. Good. So now what is batch culture? So batch culture, only single batch of media is provided. We don't remove the media. We don't add the new fresh media. Okay. So because no fresh media is provided during the incubation, nutrient concentrations decline and the concentration of waste will increase, obviously. Now, the growth of microorganism reproducing by binary fission can be plotted as the logarithmic of the number of viable cells versus the incubation time. Resulting curve has 4 distinct phases. So, I believe everyone knows about the growth curve. So, we started over here 0, 1, 2, 3 and all time of ours, and we see that there is a lag phase. Then exponential phase, which is also known as log phase. Then the stationary phase, and then we have got the death phase. Okay. Now, who can tell me about the lag phase? Why there is a lag phase? And there are several reasons for that. Yes. So before we proceed, let's discuss so that later on we can just go through what is given in the reference. Yeah. So what is the reason for the lag phase? And this lag phase time varies from organisms to organism. Okay. So what is the first reason? Guys, this is really very simple. You all can answer. Yes, start. 5 people,
Cell
5
was
reasons.
preparing.
Hmm.
Cell was preparing to divide,
So
like
when you say
increasing,
preparing means what? Like what it is preparing?
increasing size and all.
Okay, increasing in the size. Akshaya?
They adapt to the environment, like the
Yeah.
new.
So adaptation to the new environment is required. Okay. So this is the main reason, because of which the time is varying. So the organism is first, see, even you have inoculated, let's say that it was present in the nutrient agar, and then you are putting into the broth, right? तो यहाँ पे क्या होता है, न्यूट्रिएंट अगार में रख के आपने इसको कहाँ रख दिया था, फ्रिज में रख दिया था, so that the further growth is not taking place. Okay, so you have stopped the growth of that particular bacteria which was present on the agar plate में. You have got the colonies like this, right? So हम क्या करते हैं, इसको seal करके इसको fridge में डाल देते हैं so that in future we can use. Now you have inoculated into the nutrient broth. So there is a temperature difference, correct? From low to high it is going. So that adaptation, that adjustment is required. प्लस हियर न्यूट्रिएंट अगार है और आप उसको ब्रॉथ में डाल रहे हो, राइट? यहाँ पे उसका जो मीडिया था स्टेशनरी था, एंड हियर इट इज लिक्विड मीडिया, प्लस इट इज इन दी शेकर. So to adapt to all this environmental factors, it will take time. Okay, and that's why we see a lag phase. Okay, good. So when microorganisms are introduced to fresh media, no immediate increase in cell number will occur, and therefore it is known as the lag phase. Although the cell division does not take place right away, there is no net increase in the mass, okay, because the cell is synthesizing the new components. So it is actually getting ready for the division. It is, analyzing, okay, आसपास मेरे बहुत सारे nutrients present हैं. I can divide, I can grow. So they will start synthesizing new enzymes or new proteins, which is required for the division. Okay. Lag phase prior to the start of cell division, it is necessary for a variety of reasons. So it may be, let's say that the cells are old and depleted of ATP essential factors, ribosomes. So it will take time for it to start, growing all these things. So धीरे धीरे nutrition लेगा, उसको convert करेगा ATP में, and it will try to, synthesize ribosomes so that the protein synthesis can take place. Or this is one first reason. Second reason, the medium may be different. So let's say that आपने इसको nutrient broth में था. After that you're putting into some, I don't know, MS media, okay, which is used for actually your plant cells. Let's say that आपने वहां से उठा के यहाँ डाल दिया है. Though it has got certain nutrition, it will take time for the bacteria to adjust and divide. So here the enzymes would need a different nutritions. Possibly microorganisms have been injured or require time to recover. Now, how the microorganisms get injured? Shruti?
Ma'am, during the process of transfer, maybe the lack of oxygen or things like that.
No, so when we talk about being injury, the only way, it's not like bacteria को आप मारते हो, right? थप्पड़ मारोगे तो ऐसे तो होगा नहीं. So in the case of the microorganism, it is the sudden temperature change. आपने कोल्ड से इसको सीधा रूम टेम्परेचर में रख दिया. Okay, so that is like sometimes the certain bacteria might die. In a colony, there are certain bacteria, they might die. Not all, but there are chances that they might die. Or all of a sudden you're using certain acidic media or a basic media. From a normal optimum pH of 7, आप अचानक से नीचे चले गए या ऊपर चले गए, like acidic or basic. So this is like something, giving a sudden, what to say, sudden change to the microorganism that can result in the injury. Like there are not all bacteria which are resistant to all these changes, right? So it will take time for them to recover. Now, whatever the cases are, eventually the cells will try to replicate the DNA, increase in mass, and finally they're going to divide. Now, lag phase is going to vary in the length as it depends on the microorganism as well as the nature of the media. Now, this could be long if the inoculum is from the old culture. See, this usually happens, okay? कई lab में क्या होता है when you go, suppose a new person is coming and working. So we'll say that, okay, you are going to work on that particular organism. पर उस पे अभी कोई काम नहीं कर रहा है. So you don't have it from the fresh media. So it will be present in the liquid nitrogen or it is present in the -80, एकदम seal करके उसको रखा होगा. So now you are going and you have to work. So that time you can, that particular organism, you have to revive it. Right? आपने उसको freeze करके -80 या liquid nitrogen में रखा है. अब उसको revive करना है. So how you are going to revive it? So there is a slow procedure. Let's say that आपने उसको liquid nitrogen में डाल रखा है. The temperature is -196 degrees Celsius, right? आप उसको बाहर निकालोगे. Going to put into the -80 degrees Celsius. Now -80 आप कहाँ से लाओगे? Dry ice. Then bring it to 20 degrees Celsius. Then 0. Sorry, not 0, 4 degree. Okay. धीरे धीरे लाना है. So when it is coming at here, this I'm talking about minus, okay? यहाँ minus है, यहाँ ठंडा है. उसके बाद from 4, this is +4. Now you can bring it to 37 normal temperature. ठीक है, 37 भी नहीं होता एक्चुअली, आपके लैब का टेम्परेचर 25 to 28 के बीच में होता है बिकॉज ऑफ द AC and all. And then we keep it into the incubator at 37 degrees Celsius. Clear? Have you done in the lab sometimes? किया है किसी ने, चांदनी?
Yes, ma'am.
Okay, good. Okay, so that's why if it is from the old culture, so it will take time because it has been refrigerated. Now, अगर आपने इसको कोई दूसरे मीडिया में किया है, so it will take time. Let's say that it was growing very nicely on the glucose rich media. All of a sudden आपने उसको lactose में डाल दिया है. So again it will take time, and you know the concept behind the lactose. इसको पहले आपको galactose and glucose में break करना होगा, lactoperon उसका चालू होगा, and then it will start taking all this thing. Correct? Now, on the other hand, if a young, vigorously growing exponential phase culture is transferred to fresh media of the same composition, the lag phase will be short or absent. Already it was growing, so the chances is that they are not, it's not going to be there itself. So directly अपना exponential phase दिखाएगा. ठीक है? Coming to the exponential phase. Guys, clear to everyone, lag phase? Yeah, it's very simple. फिर भी अगर in case कभी doubt आता है, so you guys can ask, okay? Now coming to the exponential phase. So in the exponential phase, what is happening? So you have got the microorganism, they're growing very fast, dividing at their maximum phase, maximum rate, okay? The nature of the media and the conditions under which they are growing. So it's everything is in the favor of that particular bacteria, okay? Now, the rate of growth is constant during the exponential phase, the microorganism dividing and doubling in number at regular intervals of time. Because each individual divides at a slightly different moment, the growth curves rises smoothly rather than in a discrete jump. हाँ, ये बहुत important है. So how we have shown the graph to be like this, correct? Then it reaches the stationary and then it declines. So ये phase को हम बोलते हैं log phase. Agree? Now since each individual organism or a cell, it is dividing differently. इसका मतलब क्या? Even though it is in the log phase, let's say that 100 cells are there, each 100 cells are dividing, but it's not that at the same minute. Each 100 is becoming 200. फिर second minute में 200 से 400 होना चाहिए. Right? So this is not happening. Okay. It's like 100 का 150 होगा या 150 का 250 होगा या 250 से सीधा 500 होगा. So that growth is there. Now why? Because it is not a synchronous culture. Did you get it? Are you getting the point over here? अगर 100 बैक्टीरिया हैं तो वो 100 के 100 डिवाइड हो रहे हैं, बट इट्स नॉट सेइंग दैट at the same time, at the same moment, each bacteria is dividing. ठीक है, पहले 30 हो रहे होंगे, उसके बाद बाकी के 70 होंगे. तो जिसकी वजह से क्या होगा, हमें इंक्रीज तो दिख रहा है बट नॉट द डिस्क्रीट जंप्स. अगर जंप्स होते सो ये मुझे कैसे मिलता, यहाँ पे ग्राफ कैसा होना चाहिए था, फिर 100, फिर एक छोटा सा लैग आएगा क्योंकि सारे बैक्टीरिया अपना सेल साइज इंक्रीज कर रहे हैं, देन अगेन एक जंप आएगा, देन एक छोटा सा गैप आएगा, देन अगेन जंप कर रहे हैं, then again छोटा सा gap and then again jump. So this is how I'm gonna get if the bacteria were dividing at the same moment. Okay, यानी कि अगर मैं अभी 10 मिनट में चेक करती तो मुझे let's say 100 cells मिलते हैं, ठीक 20 मिनट चेक करती मुझे 200 मिलते हैं. So these jump I'll get. But there is a lag of 10 to 20 minutes क्योंकि उस पर्टिकुलर 10 मिनट में सारे बैक्टीरिया अपना सेल साइज इंक्रीज कर रहे हैं, एंड द मोमेंट 20th मिनट इज कमिंग इट इज गेटिंग डिवाइडेड. सो इट मींस 30 मिनट में वो फिर आपके 400 हो जाने चाहिए. सो दैट जंप आई विल गेट. ओके, सो बट वी सॉ दैट द ग्राफ इज ऑलवेज स्मूथ. डिड यू गेट इट, द डिफरेंस or understanding like how it is going to have the discrete jumps. This is important guys. ये हमने एक बार इंटरव्यू में ऐसे ही पूछा था, like what if the bacteria are dividing at the same moment, how the graph will look like. So then you have to plot the graph. Clear to everyone? Tanvi?
Yes, ma'am.
Good. Okay. So the population is mostly uniform in terms of the chemical and physiological properties. Therefore, exponential force are used in biochemical and physiological studies. Yeah. So this is the reason why the bacteria which is in the log phase are used for your studies. Okay, because they are that time metabolically active. Now coming to the unbalanced growth. So it is also a part of your exponential phase only. So unbalanced growth, it results when the bacterial population is shifted down from a rich medium to a poor medium. ये क्या होगा? For example, glucose to lactose में हमने डाल दिया, ठीक है? Now when shifted to a nutritionally inadequate medium, they need time to make the enzymes required for the biosynthesis of unavailable nutrients. It means sometimes what happens, the amino acids are there. For example, her bacteria, every bacteria is not able to synthesize the histidine, right? So they take it from the nutrient broth or nutrient media. But suppose histidine is not provided in the media, then they have to activate their operons so that they can start synthesizing tryptophan, histidine, or arabinose, any other things. Okay, so we have talked about all these operons. Now, consequently, cell division and DNA replication continue after the shift down, but the protein and the RNA synthesis slow down just because they are also needed to start synthesizing the unavailable nutrients. The cell becomes smaller in size, they reorganize themselves metabolically until they are able to grow again. Then the balanced growth is resumed and the culture enters the exponential phase. Did you get it? So it's not like the bacteria growth is stopped. It is still growing, but in a slower rate since they have to biosynthesize the unavailable nutrition. Okay? Clear? This is happening when the shift down is happening. That is, from a rich media to a poor media, you have transferred the bacteria. Now, shift up क्या होगा? From poor nutrition media to the richer media. Okay. So these shift up and these shift down experiments demonstrate that microbial growth is under precise, coordinated control and responds quickly to changes in the environmental conditions. Okay, so when the microbial growth is limited by the low concentration of required nutrient, the final net growth or the yield of cells increase with the initial amount of the limiting nutrient present. So if you know what is the limiting nutrient present, for example, in few cases it is a tryptophan or the histidine, then the growth of the bacteria will slow down. It is not stopped but slow down. Okay, and then it is going to reflect the changes in the shape of the curve. So at sufficiently high nutrient level, the transport systems are saturated and the growth rate does not rise further with increasing the nutrient concentration. So here you can see the effect on the nutrient concentration. So in this case, it is the effect of the changes in the limiting nutrient concentration on total microbial yield. So what is happening at the sufficient concentration here, the maximum high concentration, the total growth will plateau. क्यों? क्योंकि सारा saturate हो चुका है. We are not taking further nutrient concentration, right? Now what happens to the growth rate? It will become a saturation over here, right? So it will stop there क्योंकि हम इससे ज़्यादा nutrition लेके कुछ कर नहीं सकते हैं. It has reached its maximum growth rate, and that's how your growth curve is looking. So both the graphs are clear to all of you?
Yes, ma'am.
Yeah, good. This is important. Now coming to the stationary phase. So let's finish this also. Now, eventually population growth ceases and the growth curve becomes horizontal. The stationary phase usually attained by the bacteria at around 10 to 9 cells per ml. So this is when the OD becomes, more than 1. In the stationary phase, the total number of viable organisms remain constant. Okay, this also I should mention that whenever you're taking OD in the starting, we take it at 600 in the starting, or we say when the bacteria is in the exponential phase, we go with the OD of 0.6 to 0.8. ठीक है, ये आपका exponential phase या log phase determine करता है. If OD is becoming, say 0.99 या 1, that is your stationary phase, and beyond that we do not take the culture. अगर 1.5 या 2 दिखा रहा है, it means the cell is already in the stationary phase and certain bacteria might have been dead also. अब organism मर भी जाएंगे तो भी वो ओडी देंगे, right? Because it is based on the number of the cells present, right? So that's why we don't take OD beyond 1. The total number of viable organisms remains constant in the case of the stationary phase. Okay. Now this may be from the balance between the cell division and the cell death. यानी कि जितने divide हो रहे उतने आपके cell death भी हो रहे हैं. So that's why stationary phase में the number of cells are going to be same. Now, the population enter the stationary phase for several reasons. First, of course, the nutrition limitation will be there, right? So population growth will slow down, or the oxygen is not available. Since it is not very soluble, it may be depleted. So kaise rehta hai? On the top of the culture, okay, where the oxygen is directly touching the media, there you will see that the growth is present. Below, like in the deep media, the growth will be depleted. Now the cells beneath the surface will not be able to grow unless the culture is shaken or aerated in another way. It will also stop due to accumulation of the toxic waste. So kaise hota hai? Bacteria, they keep on dividing, and it results in the metabolic waste, right? So those metabolic waste are getting accumulated in the media itself. Even that can result in the growth of that can result in the slowing down the growth of the bacteria. Now why? Because when these toxic waste are accumulated, what happens? It is becoming more and more turbid. Turbid hoga, oxygen kam ho jayega, right? And the nutrition which is supposed to be available, that is also getting depleted. So nothing is in the favor of the bacteria. Hence the growth will stop, and eventually later the bacteria will start dying also. For example, streptococci can produce so much of lactic acid and other organic acids from the sugar fermentation, which is present in the media, and it becomes acidic. So what will happen? The growth is inhibited. Okay. Now, these cultures can also enter stationary phase due to depletion of their sugar supply. Now, the stationary phase they can also enter because of the response to the starvation. Starvation होगा ही जब आपके पास nutrition नहीं है, correct? So this often occurs in nature as well because in many environmental cases, places they don't have the nutrient availability. So sometimes the starvation can be a positive experience also. This is important, guys. Now, why? Because this will lead in the production of new starvation proteins. Okay, and this will make your bacteria more resistant to the damage. Okay, so these starvation, it is actually a positive experience for the bacteria, making them more resistant to the damage. So they make their cell wall more thicker, they increase the strength of the cell wall. There will be more increase in the cross-linking in the peptidoglycan, because of which the cell wall is becoming more resistant towards the harsh environmental factors. Okay, so eventually it is protecting the DNA. So the DNA binding proteins from the starved cells protects these proteins, they are going to protect the DNA. So this is important. Okay. Then there are chaperones. There is increase in the production of the chaperone so that their protein can be prevented from the denaturation. And as a result of these many other mechanisms, the starved cells becomes harder to kill. Okay. So what happens is that even if you now add certain, agents like antimicrobial agents, since the cells have become so hard, like so resistant, they will survive. And these bacteria can survive starvation for many years. Like एंडोस्पोर फॉर्मेशन में चले जाएंगे, या they will form a mucin layer so that they can protect themselves from the harsh conditions of the water or the pH, so they will keep themselves protected. ठीक है. Now what happens is that in future when they get a proper nutrition available, then only the bacteria will start growing. Okay. Now coming to the last phase, that is a death phase. So death phase, as we know, nutrients are not present, toxic waste is getting more accumulated, and now the cell will start dying. Okay. Often the only way of deciding whether a bacterial cell is viable or not is you take from the death phase and put it into a new media. If it is growing back, if the media is becoming again turbid, it means the cell was viable and it has grown. If it is not growing, then we say that the bacteria is totally dead. Okay. Now coming to the loss of viability. So here either you can go for the starvation or the growth arrest. So in the death phase, you have very few cells will be there which is remaining alive. So when you put into a new media, you can see that only few cells are growing. ठीक है. कुछ होते हैं जो genetically programmed होते हैं. So they are like कुछ बैक्टीरिया ऐसे होते हैं, they will undergo suicide so that their brother and sister can survive. So they themselves will kill, and उसके मरने से जो nutrition release होगा, on that nutrition the other bacteria will be surviving. ठीक है. Then we have got the genetically programmed sterility or the viable base number of their cells. So इसमें क्या होगा? It's like only the resistant bacteria they are going to survive. Okay, whenever there is a change in the media or any change is there in the environment, only that particular cell is going to survive. Okay, so this is how you can do in the lab the check. So there are three different ways by which it can be done. Yes, guys, any doubt in this? So I'll be stopping here itself, and in the next lecture we'll be completing this topic where we are going to discuss like the effect of pH, temperature, and what all. So in the next lecture it will be over, and that lecture will be today itself. I hope everyone is having leave today. It's Saturday. Yeah. So can I keep it around 3
ma'am.
o'clock? No.
No.
Achha, who is it? Shruti. Shruti won't be free, right?
Yeah, no, I can't.
Okay. Okay, if I keep it at 7 then?
Yeah, understand.
Okay. So, okay, then at 7 another topic so that we can